Okay, let's get going. So we're really excited today to be able to bring to you Stan Lapidus, who's a well-known entrepreneur. And as we get going, let me just pop forward into a couple of slides. So what is Medical Device Playbook? We've been running this event for about 6 years. And for the first 5 years of its life, it was just like this in this video, it was a full-day conference. We held it in 2 cities, in Vancouver and Toronto, and we brought speakers from all over North America, and it was networking, all the traditional things. Well, guess what happened at the beginning of COVID? So we started to have to go virtual, and we decided since we were going virtual, we'd make it shorter and we'd make it more often. So we actually have been running them every month, and we've run a number of them. If you're interested in looking at them, go to our website, starfishmedical.com/webinars, and you can see a number of the other webinars we've run on all sorts of interesting, useful topics. So anyways, so we're also finding that we now actually have almost twice as many people signing up as ever came to the live conference. So we're delighted to be able to offer actually more impact to entrepreneurial companies. That was really the deeper intent. How can we inspire people in entrepreneurial companies to make the tough choices they have to make, to start the right companies, to design the right value propositions, overcome all their technical and market challenges, etc. So that's what Playbook is. A little bit of context about who we are. I'm the CEO of Starfish Medical, which is a medical device services company. So let me tell you a tiny bit about that. So we have 2 main offices, Victoria and Toronto, serving the North American market with about 150 staff. So that makes us one of the largest companies in our space in North America. We also have representation in California and Boston, and our clients are all over the place. So what sort of companies do we work with? A mix of large corporates, you maybe recognize a few of these corporate type companies, and startup type companies. It's roughly an equal mix, and but we think of them all as entrepreneurial. They're all wanting to do something novel and exciting that will transform their space in some way. So, and what sort of things we design? Things that we care a lot about how they work, and we care a lot about what they look like and how they interact with people, and particularly we care about the value proposition. I know that we're talking about some of that with Stan. So things like ingestible robotic assay platform. Nobody had ever heard of such a thing before we worked on that. Or dynamic light scattering for platelet quality, or assay platforms, or blood flow fluorescence for plastic surgery. Those are the kind of. So let's get into things here. And let me talk to you a tiny bit about Stan. I should mention that there's still a lot of noise in the background here from somewhere. So if in your interface, that you'll see that there's a Q&A tab. So go ahead and stick any comments or questions you have in mind there, and I'll try to keep an eye on that. Given that this whole event is a Q&A format, then I'll just try to sprinkle in and keep tabs on what those things are. So let's move on to Stan himself. So Stan is a very distinguished entrepreneur and engineer. He's currently the chairman of the board at BinX Health and Mervy. He's on the board at Path AI, at Glimpse Bio, at Fractal Labs. He's a founding pillar of Pillar VC in Boston. He's got his own Lapidex research incubator, and he's an executive in residence at the University of Colorado. But mostly what we want to be talking about today, he's been the founding CEO of 2 companies that both went on to become $1 billion in the diagnostic space, and both totally different. So there's Cytyc Corp, which he founded in 1987, which revolutionized early detection of cervical cancer through this development of the modern Pap test, the thin prep. In fact, one of the 2 of his prototypes are at the Smithsonian, so that gives you some idea of the impact. Exact Sciences, which he founded after, pioneered non-invasive early detection of colorectal cancer, so has become the fastest growing test in the history of the diagnostics industry. So that's pretty remarkable. Hold 37 patents, primarily in cancer detection. a fellow at the American Institute of Medical and Biological Engineering, as an instructor at MIT. And 51 years ago, he graduated with a Bachelor of Science degree in Electrical Engineering from Cooper Union in New York City. So, welcome to the show, Stan. It's a total honor and pleasure to have you here with us.
Thank you, Scott, and thank you all for your participation.
Great. So let's roll the clock back to your early career and just tell us a little bit of circumstances of kind of what led up to the Cytyc founding and what made you think that was the right thing to do.
So I have, as Scott mentioned, a bachelor's degree in engineering. As most people do with bachelor's degrees, I started a job. I worked at, in the 1970s, I worked at Raytheon Medical Electronics. Raytheon is primarily a defense contractor, but they had a small nuclear medicine business. And I wasn't involved in the medicine. I joined because of my interest in the interface of weak signal nuclear physics and electronics. The principal product of the company was a gamma camera. It didn't work well, it drifted, and all attempts to correct the drift were unsuccessful on the part of physicists much smarter and more accomplished than I, not just at Raytheon, but across the industry. At Raytheon, the problem was acute because Raytheon had the largest gamma camera. It had 91 photomultiplier tubes, so they would drift out of balance in a day or two. So I invented my first patent, a simple device which measured the non-uniformity of the gamma camera when exposed to a point source of radiation, and did a simply 1/n calculation. If a spot on the image was too hot, we made the pulse width narrower. If it was too bright, we made it narrower. If it was too dim, we made it wider. So that actually, in the nuclear medicine industry, is not very large, but it was the first of its kind, and it did revolutionize that industry. It enabled the development of follow-on products like PEC-T. I got my first patent. Raytheon paid me $100 for that instead of the $1 it had to by statute. And I got a raise from, this is the 1970s, from $25,000 to $32,000, a huge raise. And I was despaired. And I thought that if I can invent this for Raytheon, I can invent many things and I can do it on my own. So in 1978, I quit my job and started an engineering consulting company with a college classmate of mine. And we had no specification as to what industry. Most of our work took place in factory automation, developing industrial controllers. And one of the products that we saw a need for, that our customers told us about, was industrial machine vision. So we spun out from the consulting company, our first venture funded company. I raised, I think, initially $750,000, eventually something like 9 million, if I remember the numbers. And it was very difficult. We had 200 competitors. We had one patent that was useless in keeping the competitors out. So we had a huge market. And we also had a very vague value proposition. Our story was we focused on the automotive industry. Our story was we would provide higher quality product that would go into the cars, parts that would go into the cars. We would cull out defective parts before they were built into vehicles. The problem was factory managers at General Motors, which was our principal customer, weren't paid for producing high quality cars. They were paid for producing cars. They had no incentive to adopt quality control technologies as measured by the bonus. So the CEO would go off and talk about the quality initiatives at GM, but the bonus checks were based solely on productivity. So every sale was hard. So it was a 3-year, 4-year slog. At the end of it, we sold a company it merged with a public company for about a $20 million valuation. So the $9 million that went in meant that everybody made money. The founders did, the investors did, but it wasn't a lot of money. And for all the hard work that went in, each of the three of us were disappointed. Two of the investors, of the founding investors of the company, it was called iTran, approached me and said, Stan, we don't know what you're going to do next, but here's $200,000 and go figure out what you want to do. And if we like it, we'll put more money in. my thinking was that I should, my thinking was actually initially how angry I was at my two partners. We didn't make a lot of money. It must be because they're jerks. I'm sure they thought the same thing about me. You know, we worked very hard, little to show for it. And so I began thinking, rather after I stopped thinking about, gee, these guys were jerks and it's all their fault, which it wasn't, of course, is that really the problem is that we didn't have a good business. The fundamentals weren't very good. And I wrote down a credo, and it's something I still practice. I try to practice these days in companies that I found or in companies whose boards I join. And the elements are huge markets, clear value proposition, strong IP. I touched a little bit upon the shortcomings of ITRAN, why the market was huge, our value proposition was weak, and our one little patent didn't defend us against anyone from competing with us. So with that in mind, I started a broad survey of machine vision applications that could replicate, that would not be done, you know, we wouldn't sell 10 machines to GM for a given application. We could do thousands and tens of thousands of machines, and quickly stumbled across problems of imaging Pap smears. Pap smears were invented by George Papanicolaou in 1928. It took him about 30 years to see that his initial idea was commercially successful, and commercially successful was measured at a population level by how many women got Pap smears. Well, the more women that got Pap smears, the more rapidly the decline of cervical cancer became. Initially, cervical cancer, from the beginning of time until the 1950s, cervical cancer was the #1 cancer killer of women. Today, it's breast and lung cancer, very different epidemiology 3 generations ago. The great success in the Pap smear, and I'll explain in a moment what it is, points to an epidemiologic measure. Do something big, do it in a way that you can measure it, and that may well lead to better outcomes than we had at ITRAN. The Pap smear is made by collecting cells once a year from a woman's cervix. By the woman is in stirrups, the cervix is everted, and a little either wooden spatula or plastic broom is used to smear, or not to smear, to sample the cervix, and the results were smeared on a slide. The smears were full of blood, mucus. They were often inadequate and had to be redone, which was very expensive, and not all patients complied. But even so, there was a marked epidemiologic decline in mortality. To me, this was a rife problem for machine vision. I thought that the constancy of machine vision would reduce false negatives. The general feeling was the false negative rate of Pap smears was about 50% or so. So we came up with a machine vision system. We started to work and spent the $200,000 and raised a bit more venture capital. And the scary thing is that the results, we could find cancer cells, but we weren't finding them as well as humans. This is with Apple Macintosh circa 1987 now, technology. And this was looking pretty grim. So really quite out of desperation, on a Saturday afternoon, I came up with an idea for making a better Pap smear, laying cells in a thin uniform layer on a slide. So this was an invention born out of desperation, which is often how inventions happen. The idea was to use a plastic cylinder with a taut polycarbonate membrane across it, which had holes in it. We'd suck up cells onto the filter. When the filter was 10% covered, we'd pull the filter out of the collection fluid and press it onto a slide. So a very simple device, an electromechanical device. And after initial clinical evaluation, we were astonished to see that the pickup rate of cancer and precancer was higher on the ThinPrep slide, that's what we called it, than on the conventional slide. Not that it was something that a better pathologist could find had he or she only spent more time on the slide. There really were more abnormal cells, and there were many cases which showed up on the ThinPrep and not on the conventional slide. And in retrospect, the answer was simple, but we didn't know it at the time. We spun the filter in the fluid, which caused a uniform sample, subsample of what was on the cervix to get onto the slide. In making a manual smear, this smearing process doesn't assure that the cells are uniformly distributed. So we had something that was better. It took us forever to get through the FDA. They had never approved a cancer screening diagnostic test before. It took over 5 years. But after approval, the test began a very nice rollout. This was in 96. In 2002, by 2002, the company was declared a monopoly by the Federal Trade Commission. And the reason is shortly, a few years after the initial approval for equivalence to the traditional Pap smear, the FDA awarded the company a label of superiority. With a label of superiority in a field in which there was a lot of litigation, Cytyc became the standard of care, and anyone who was using the conventional Pap smear was practicing substandard medicine. So the switch became pretty quick. And from 2002 until the company, went public in 96, in 2002, until the company was acquired for $6 billion, sales grew steadily at about 25% a quarter, which is to say doubling every year. And the exit came in 2007 with the $6 billion cash acquisition by a merger into Hologic. This is a story that took, therefore, from 1987 to reach its endpoint, 2007, 21 years, created a new standard of care, based and became a big business and became a hallmark for gross margins. The company had years in which gross margins were 90% like pharmaceuticals based on the value proposition, and earnings before taxes were over 50%, again, based on the value proposition. So the things that went especially right in retrospect were, I think, identifying a big problem and developing a unique solution which was well protected by IP. Along the way, I transformed from a guy who was a working electrical engineer, designing circuits, writing firmware, into a guy who I actually, along the way, picked up an appointment as an assistant professor of pathology at Tufts Medical School, the only non-MD, non-PhD. I got sucked in by the medicine. Long story short, I acquired a new vocabulary and a new framework about how to think about value. And value looked at through the eyes of patients, physicians, and payers. Those are the three elements of value. And what I learned was it's important to be able to measure value in cost per procedure, in reduced callbacks of the patient because of inadequate or suboptimal Pap smears. And finally, though it takes a longer time, through metrics of continued reduced mortality, which is indeed now clear that the ThinPrep contributed to that. So that's the story, Scott. That's the Cytyc story and the story of my journey.
That's quite a story. So I always find this fascinating. So there's any number of things that are fascinating in this. One of them is actually the first thing that you said that caught my ear was when you talked about your first automotive company and you're trying to sell to plant managers who are not actually incented by the overall system good, which actually is amazingly analogous to what happens in medtech, right?
Yeah, exactly.
That actually good health outcomes of patients are not actually a financial outcome for the system. So we're all kind of stuck in this never land, right?
That's correct.
And so that's other than that observation, I think that's maybe something we don't need to talk about in depth. But I was interested in this spot where you have this company, have this premise, hey, we can use Apple Macintoshes or whatever, the computers of that horsepower to find these misshapen cells, and they'd be better than people. And then it turns out that premise wasn't true. So there must have been some dark days in there where you had to raise capital, but at the same time you were challenged. To walk us through that time, because that's actually the dark night of the soul of entrepreneurship, right?
I think most of my days are dark, to be clear, as a working CEO. Now that I'm a board member, I don't have dark days. The other guys have dark days, And if it doesn't work out, we fire the CEO, we change direction, but it's a very different life. But as a CEO, as a founder, and as an inventor, and any given day, nothing is going well. And this isn't the operational, the factory has a quota, didn't hit the quota, the sales guys have I'm not talking about that. I'm talking about the technology isn't working. And I think my lasting lesson here is it simply takes the resilience to emotional resilience to live with that, and the tough-minded decision-making, which is in fact something that board members are very helpful with. Like, Stan, the data aren't getting better. Are you sure you want to continue this, but just working on the imaging? And so this requires a good dialogue with board members, and board members who are clear-eyed and who aren't there just to support management, but to be critical in their thinking about how the team is doing. For us, for my co-founder Fred Farber and me, we could measure our ability to find cancer cells and compare ourselves to what we understood human performance to be. But again, we were asymptoting. It was an exponential curve to which we were asymptoting of performance less good than humans can do. And that's what really ignited the effort to accelerate what until then had been, maybe we should work on a prep, maybe we shouldn't. Prep would add cost. Prep would require retraining of the technologists, the tens of thousands or 10,000 technologists who read Pap Smear slides. So there are many reasons not to do it. But the reason to do it is without it, we couldn't get the imaging working. And what we finally learned was that we didn't need the imaging if we could make a better Pap Smear. It became so much easier to read, so much quicker to read, that the need for imaging was made moot.
That's amazing. So that's kind of a perfect example of a startup pivot in essence. This whole framework for which you started the company turned out not to be the key to your success at all. And you talk about that journey, mental gymnastics, as it were.
I remember driving, there was a lake in Marlborough, Massachusetts. I remember driving by the lake and saying on a given day, it was early on, well, let's see, the imager isn't working. What do we have to do? We do prep. Let's start a little bit of prep work. And we had this idea, but going from the idea to something working took over a year, even at a prototype level. And every day it was, this is working worse than that. I should worry more about this than that, until finally the prep pulled away. And we kept both programs for a while, and then we finally killed the imaging program, which is hard because these were people who had worked very hard. We laid them off. We had little to show for their efforts after their years of hard work. And then the prep team pulled ahead. So these are very tough on a personal level, with people with whom you've had an emotional relationship, with whom you've worked together as a colleague, the days of the long nights, as we called it. But the most important part is being clear-eyed about what's working and what isn't. And this isn't something that I think I do by myself. It's something that our team does and our board does. And not all board members are of equal value. I was very fortunate in working with a guy in both Cytyc and Exact named Ed Kenya of Flagship Ventures. And he and I would speak probably 4 nights a week, 1 to 2 hours a night for, 10 years, first at Cytyc and then later at Exact Sciences. And Ed's role as a board member, he was very good at this. He was careful about not moving into the CEO's prerogatives. He never said, Stan, do this. Everything would be couched in the form of a question, and the questions would be quite deep. And I think working with Ed in particular, and my co-founder Fred Farber in particular, helped us execute the pivot well. So a pivot is about a team. It's not just about, I learned, it's not just about a solo guy, which was, I should tell you, my orientation prior to this pivot, because I thought of myself as a solo inventor and the team was there to support my inventions. This is very different. When you're running a business and you have tough decisions like pivots to make, you really want the perspectives of others, people who are invested less emotionally than you are, who see things more clearly.
Mm-hmm. So tell me a little bit about this 5-year journey through the FDA. That must have been some very long nights of the soul as well, as you,
yeah.
That's probably not what your plan was, that you'd raised your money with.
What happened was, I'll tell you the long version of the story, because the point is that everything happens in a context. In the, I forget now, late 80s, there was some scandal at the FDA about 2 out of their thousands and thousands of employees, 2 employees were bribed by a company to either expedite a clearance or clear something that shouldn't have been cleared. I don't remember the details, but the long story short was those guys went to jail. The FDA commissioner was fired, and a new guy was brought in, and he was the new sheriff on the block. His name was David Kessler. And his view was that the device side of the house, as opposed to the drug side of the house, was in total chaos. Everyone was a crook, and they shouldn't be trusted. This is his own employees. That is, there was a presumption of guilt. That ground the agency to a halt. There was a period of time in which you couldn't have a phone up, have a conversation with a reviewer. There had to be 2 people on the phone, because Kessler felt that if 2 people were on the phone, the opportunity of proposing a bribe, that's the way he thought, would be much reduced. Scheduling 2 people plus the company became much harder, and the pace of everything slowed down. He finally left because he himself was caught up in a corruption scandal. He cheated on his expense reports, no less. He went on to become the dean of Yale Medical School, from which he was fired, dean of UCSF, from which he was also pushed out, and nevertheless survived somehow in the Biden administration. He's a cat with 9 lives. So he's a guy that I think of as a zealot, and zealots in government aren't helpful. That's the background. Mm-hmm. The actual story is, when I had the 200K, but not yet started to raise the millions, we went to the FDA, and they wrote us a letter that if you proceed with the product you've described, we will approve it with a 510K, which you may know is a much less costly process by about $90 million than a PMA. So based on this, I raised startup capital. I actually built a factory. We made our 510K submission. And during this interval, from the time I started the company till we made the 510K submission and had the letter from the FDA saying you can do a 510K, Kessler started to blow up the agency and tear it apart. The upshot was, I remember walking down the stairs after we made a presentation to the FDA, and one of the branch chief, as he was called, said to me, "Stan, I'm afraid this is going to be a PMA, but I have in writing from you this is a 510K." You can tell I'm still bitter after this 30 years later, because generally in life, your word is your bond. If you put it in writing, it's more than your bond. And here was something that had ramifications that were life or death for the company. We made a decision to use the data that we had submitted in the 510K, which wasn't as carefully audited as it should be, but it was still legit data, but we didn't have the paper audit trail because 510Ks didn't require it at the time, and that was a mistake. So we submitted the 510K, we had a successful panel hearing, the company had to redo the PMA with a new data set, and by the time the journey from the 510K to the PMA was done, 5 years on the clock had elapsed for no purpose, because the product was the same and the results were the same. Do I sound bitter, Scott?
Well, appropriately so. Most companies would not have survived 5 years of twists and turns. Somewhere along the line, the investors would refuse to put more capital in.
Yeah.
So,
well, this is an important point. So this is about as bad a situation as you can have with the agency. And the company survived because the value proposition and the size of the market justified continued investment. And there was one down round, but it was all justified by the size of the market. So had we had a smaller market, or had the value proposition not been as compelling, I think the company would have folded.
Mm-hmm. Yeah, amazing. So I'm interested in a few things here. So you talked about IP, and this is maybe a good opportunity to segue into IP and what your strategy was at the time, and how'd that work out?
Yeah. So we had the insight that we had. So people had been working. So I don't want the audience to think that the idea of making a better Pap smear was an idea in a vacuum. George Papanicolaou had first started with liquid preparations himself, and that had been superseded with the smear. He didn't actually invent the smear. A guy named Ernest Ayer did, because it was easier to make. Papanicolaou irrigated the vagina with a bulb and then had a hard time getting the cells from the bulb onto a slide and making them stick. So people had used various methods of centrifugation, other prep techniques, not without much success. when the requirements of screening medicine are, it has to be highly reproducible, it has to be cheap, and it has to be able, at least as the problem was defined then, to be performed in a physician's office. So liquid prep approaches had been around for a while without much progress. So we had kind of read everything, met most of the people that were still alive working on this, and thought all of these approaches were nuts, that they were too complicated, required too much manual effort, and were too variable on how to make a thin, uniform layer of cells on a slide. So we thought we could carve out some pretty good IP space by rethinking the process. And our principle is based, if I were to do slides here, it would show a French press coffee pot and make the point that as you press the plunger down, the pressure differential across both sides of the filter in the French press coffee pot is proportional to the area of the filter that's covered. That's the principle we used. The differential equations were pretty simple. And we went out, I think we ended up with 7 patents, not a huge portfolio, but with pretty broad claims on methods of making better Pap smears. So we had good broad IP coverage. I want to go back to a story I told you. This is a patent story. So I mentioned that at Raytheon, I had invented a way of making gamma cameras operate more uniformly, and the company filed a patent on my behalf, which was the worst written patent I had ever seen before or since. This is some hack lawyer at corporate headquarters who couldn't get a job at a law firm, I imagine, so he wound up at a corporate patent department. And it blocked no one. So the company had 11 competitors, eventually all 11, in one way or other. They didn't infringe because the patent was poorly written, but had the patent been well written, they would have infringed. So I learned the lesson at Raytheon that you have to think only, we as engineers, as scientists working in healthcare, have only 2 questions to think about. One is, can I practice freely? And the other is, can I block others? Our job, I believe, is, I know very often inventors write their own claims and write their own patent disclosures. I highly recommend against this. What I recommend that, because we're never going to be as good as the best patent lawyers. What I do recommend is that we inventors, we engineers and scientists, think as broadly as possible about that which we have invented, discuss with your patent counsel, as I discuss with mine. Tom Myers is the guy I've worked with now for almost 30 years. Tom, I think, I've discovered this, and he almost always comes back and says, no, Stan, this is an instance of what you've invented. What you've really invented is this. And by filing broader claims in a tiered way, you'll be able to block others and ultimately protect your invention in a way that's meaningful to healthcare outcomes and meaningful to the business. So along the way in my journey from Raytheon to Cytyc, because I had no patents in between that were meaningful, this idea of thinking about breadth and working with patent counsel who understood how you block others became central to my views on IP.
Brilliant. So we often talk about how engineers need to really understand the ideas of IP and do their own background research and understand generally how to structure their thoughts around IP and then consult with the IP attorneys. But I think a lot of people have the idea, they describe their invention to an attorney and then they think they're going to get good protection out of that. I don't believe that is true.
That's not right, Scott. I agree with you. That's not right. What you'll get is a poorly written patent that will block no one, and you may be sued because the background work wasn't done to make sure that you have freedom to operate. So I spend a lot of time on IP, but not writing out the claims or writing the specification. Spend most of my time reading, making sure that I understand prior art. And that means reading 2 literatures. One is the patent literature, and the other, which an engineer reads differently than a patent lawyer, I should say. And the other literature is the scientific literature, which not all patent lawyers read deeply. So you have to do both. And that's something we as inventors can do at least as good as patent lawyers and frequently better. But in terms of thinking through the implications of what an invention is, making sure that your IP counsel reassures you that the breadth of what he or she is filing is the broadest breadth that's possible. And a good litmus test is if you, this is from my own experience now, if you think you invented this, if your lawyer says, great, I'll write it up, he or she's not the right lawyer. If you say, I invented this, and he or she says, no, you invented this, that's the person you want to hire. That's a pretty simple litmus test. And in a 1 hour discussion, you can figure out if he or she is the lawyer for you.
Yeah, I totally agree. Brilliant. So let's branch that slightly, or go back to that point you made about academic literature. In my experience, most technical founders don't actually read the academic literature. They've got so many other cats to point sticks at that they just don't. So I'm curious the discipline that you developed to continue staying in touch with that literature.
Well, it's a great question, Scott. Remember, I started with no background in medicine. My nuclear medicine experience doesn't count because, again, I was working as a physics geek, not as a circuit designer, not as a guy enmeshed in the medicine part of nuclear medicine. So I had a lot of reading to do. I spent out of that $200,000 of startup time, much of it went simply to fund my reading time at the library and buying textbooks. I remember going on vacation with Ruth and the kids with a medical dictionary in my lap and a textbook on the desk. I'd go 4 hours a day to the British Museum library and not be a tourist and read. And that reading carried over when the invention stuff came. So it was, for me, it was a more seamless process. And I think to the extent that what Scott says is true, I think that being systematic about spending a few hours a week reading in your field, mostly these days it's reading abstracts online, you don't have to crack papers, but making sure you understand not just of who's doing what now, but who did what. Well, patents have a 20-year life, so 20 years ago. I went back to the 1928 stuff and found the first slide that Papa Nicolau ever showed of what an abnormal cervical cell looks like that's pre-malignant. And that's what I'm trying to do is convey to you that a literature search mostly is recent, but there's value in going back deeply as well. That's where I learned that he started with liquid preparations and it transformed into something else that was cheap but suboptimal, and we reversed that.
Amazing. So you went on vacation to England and you spent 4 hours a day in the library reading technical papers, and you stayed married. That's a very wonderful accomplishment.
Well, Ruth's been wonderfully tolerant of my career ambitions.
Brilliant. Okay, let's start. We have to wrap up on the Cytyc story. But can you talk a little bit about your technical journey and how far along did you stay CEO, and what did you need to learn, and maybe a little bit about that?
So the story of my pendency as CEO is tied into the story of Exact Sciences. So quite prematurely, as it turned out, but we entrepreneurs often move on to the next product and say, well, this thing sort of has legs. We were in the middle of FDA hell, but that was unclear yet. And let's think about other applications, not knowing that it would take years more to bring the Cytyc thing to fruition. So I started to think about what other areas of medicine that had not been addressed by the discipline of cytology, the study of cells on a microscope slide, could we extend our technology to? We knew a lot about working with samples which were full of blood and mucus. We understood the epidemiological implications of epithelial cancers, how they develop over perhaps a decade from a pre-malignant cancer into a cancer. And that narrative was pretty clear to us. My CFO, Tom Farb, and I went to lunch, and he was very good at asking tough questions. And we had good valuations in the venture community. Tom would say stuff like, he did say at this lunch, why is it that our valuation is so high? If we're a lab equipment company, microtome companies aren't valued highly, staining companies aren't valued highly, we are. And the answer is, so I said, Tom, it's either because we're a women's healthcare company or we're a early cancer detection company. And we started playing back and forth with both of those ideas. And the question arose, if we're in a cancer early detection company, what other cancers are there that are epidemiologically important for which cells might be accessible that we could make a difference and have a strong value proposition. So the most you may know, the most deadly cancer of all in Western countries is lung cancer, caused 80% of which is caused by smoking. And cells of patients who have smoked for many years are coughed up in the deep coughs that smokers make. So there's some evidence that cancer is there. But by that year, which was, I don't know now, 94 or 93, it was clear that early detection of lung cancer actually did not save lives. Later, through work 2 decades later, it saves a small number of lives. But back then, there was clear medical evidence from reading the literature that if you take sputum samples and you look at the cells from the sputum and try to identify cancer, which others had done before, in prospective clinical trials, mortality actually increases. If you find cancer cells, the test turned out to be nonspecific. This is not our work. This is the work of others with a clinical trial at Sloan Kettering, Johns Hopkins, and a third institution, Sloan Kettering, Johns Hopkins, and Mayo Clinic, that early detection of lung cancer led to surgeries. The surgeries have a high mortality rate because these people don't have great lungs to begin with, and cracking the chest is never something you want to do without strong evidence. So long story short, cross lung off the list. The next most common cancer is colorectal cancer. And the colon, which is way more common than cervical cancer, to be clear, it's the deadliest cancer in non-smokers. It was 25 years ago. It still is now. The cancer is an epithelial cancer. It grows physiologically and topologically outside the body. There's a basement membrane that protects the rest of the body from those first cancer cells that grow as an adenoma, a precancer, just like the cervix cells have a precancer. Take out the precancerous cells and you've cured the patient. This is not palliation, this is a cure. That's why the Pap Smear is so successful, because it finds precancers. And we thought we could do, completing the metaphor, that if we could collect cancer cells or precancer cells from the colon, that we could do to the colon what the Pap Smear did to the cervix. The standard of care for colon cancer screening, colonoscopy, was a very difficult standard to comply with, and compliance has never exceeded 45% or something like that. Most patients who should get a colonoscopy don't, because it's a miserable procedure. We thought we could collect intact cells from stool, that the stool would sample this whole colon, some small fraction of them would be pre-malignant. We could find that through microscopic examination and prepare the samples using a thin prep-like device. We had worked with blood and mucus. This adds fibrous material and bacteria. We thought we could develop, so it's an engineering problem, how do you work with stool in a way that's not offensive to the patient, to Federal Express, to the lab? We thought that's an engineering problem. And we got very excited. We did some research. We found a guy who had published, a scientist at the Department of Agriculture in the US, who had published on this one paper. We tried to replicate his work. We couldn't. We hired him as a consultant to have him replicate his work for us. He couldn't, so we figured he's full of crap. He was a liar. And that before giving up on the idea, Fred and I theorized that, well, maybe, the cells are not there because they might be lysed by stool, but maybe the DNA is intact. And we knew enough to know that two things. One is that DNA is a robust molecule. Only two things break it up. One is ionizing radiation, which is like X-rays and sunlight. And the other is endonucleases, enzymes that eat DNA. They either eat it from the middle or they eat it from the ends. Endonucleases don't exist in the colon. They are taken out of the fecal stream as it forms, largely in the small intestine. So we thought we had a good shot at it. We thought the idea was original and we were congratulating ourselves. We have an important value proposition, some IP to make, and a monster market, much bigger than the cervical market. But we learned that a scientist at Johns Hopkins named Bert Vogelstein had published on this in '92, so a few years later, before we had the idea. So it was his idea, not ours. That was the first original thinking. And then we worked very hard to get Bert to be an advisor. So long story short, I left the company, Cytyc. I started and turned it over to a wonderful executive named Pat Sullivan, who had the skills that the company needed going forth. He's a commercialization guy, and started Exact. I raised the money for Exact actually very quickly. Now I was already a medical guy, and Cytyc gave me some brand value, and I thought, okay, we've learned a lot of lessons, and we're not going to make mistakes of the past. And somehow it still took 19 years to become a success, through the whole process. It took far longer to develop the assay. It took the FDA process was very smooth, but it took 3 iterations of the assay to get it right. 4 CEOs. I stayed on as chairman for quite a few years. And the 4th CEO, Kevin Conroy, is the best CEO I have ever met. And he took the company, and he's the guy that made it the fastest growing company in the history of diagnostics.
That's a remarkable story. So just to put a bow on the Cytyc thing, then, so you were the CEO at the time, and you were starting to think about your next thing, and presumably you're also thinking the company needs somebody different from me in the next stage.
Yes, that's right. That's right. And I think an important lesson, I had developed a very clear idea of my strengths and weaknesses, in part because the board's good at holding up a mirror to these things. I'm a pretty good engineer, not the best I've ever met. I'm an ordinary executive in terms of running day-to-day business, and I'm above average, maybe highly above average in this inventiveness stuff and seeing things and tying together unmet medical needs with ideas. So I thought that the best personal business plan for me is maximizing that. And so when I had achieved a level of something, time to bring in a CEO. And I did this in each company from the outset with the board's understanding that there would come a time that if they don't fire me, I would tell them when I was ready to go. And that's how things have played out. This is a hard thing to do, but not each of us is Steve Jobs. I'm not Steve Jobs, who turned out to be very good at running a large business, better at running a large business than he was at running a small business, far less chaos in his second act at Apple than there was chaos in his first act. I would have simply led the company into chaos.
Right. Interesting. So as sort of an innovator, presumably, I mean, you're very good at lateral thinking. You're just a continual churn of new ideas. How did you learn how to be effective at operations, or effective enough to keep the wheels on?
Yeah, keep the wheels going. I would say a very strong board and a culture of honesty, a candor among the team. So the team would be quite ready to tell me when I needed to step up my game, the team that I had hired. So those are the 2 elements.
Great.
But to be clear, I don't think of myself as an especially differentiated executive.
Well, you're differentiated on your results, so something is magic there. So it's that's remarkable.
And
so
part of it is yeah, and part of it, so that, something I'd like to communicate to the group. Each of you is attending today's webinar because you believe in your strengths. I think for real success to take place, one also has to be cognizant of one's weaknesses, and some people have none, like, well, Bill Gates, who built an amazing company from startup to what he did, perhaps like Zuckerberg, perhaps like Sergey Brin and Larry Page, though they bought in, quote, professional management fairly early on in the Google process. So each of us has to be, I think, proud of what we're good at and sufficiently modest about what we're not good at to facilitate change when needed.
Yeah, I think that's a wonderful subject. I'm glad we were able to talk about that in the context, sort of a real clear-eyed view of kind of where your energies are best spent. Can you talk about the earliest days? We have about 9 minutes left here. The earliest days of Exact, and obviously a lot of waffling around. What were your early capital raises? What were the stories you were telling in the early days, and how'd that go?
Right. So we funded the company with Flagship, Ed Kenya's firm, and I were the founding investors. I think Ed took 2/3, I took 1/3. Within it was $180,000 or $200,000, one of them, I don't remember. Within a year, we had enough bench work done. I had brought in my co-founder, a guy named Tony Schuber, who's one of the great molecular biologists. He was a mid-level functionary at Genzyme who didn't have a PhD, and he was treated like dirt because he didn't have a PhD by his boss and his boss's boss and so forth, because Genzyme is a science company, and I guess you're not a scientist if you don't have a PhD. So I bumped into Tony and made him an offer he couldn't refuse, which wasn't very expensive because he was treated so shabbily at Genzyme. So Tony and I began bench work, me kibitzing, I should say, and Tony on the bench. And within a few months, within 6 months, we had proved that we could extract DNA from stool, and that we could get it in sufficient quantities that if cancer were present, we could detect it. These weren't cancer samples, these were normal samples. Our family members were producing samples for us in little buckets. We had developed a prototype gizmo with my buddy Walker Sloan. We developed a way to homogenize stool that didn't require doing this and sniffing it or anything, so an industrial process based on a paint can shaker, industrial paint can shaker. That was the idea. And that was enough money to get 2 A-list venture capital firms, Greylock and Highland Capital, to put in $750K. And after that, we were off to the races, Scott, and we always raised money. I took the company through the IPO, and then was chairman, as I mentioned, for a few years after that, 5 or 6 years after that, and then I was on to my next things. But it was those first experiments that Tony drove that made the next tranche fundable. The fact that Flagship was involved and that I had put my own money in made accessing A-list investors pretty straightforward. And after that, the thing went to the moon. You know, despite ups and downs in the business, the company's financing history was strong. And then Kevin has shown the rest of us how you really raise money at scale, you know, doing billion-dollar financings, none of this $12 million Series B kind of stuff.
Great. And do you recall what the early valuations were that you were justifying?
Well, I do recall this because it's a story I often tell. So as I mentioned, I ran the company through the IPO, the completion of the IPO. And we went public at, I think it was 200 million pre-money, but we funded the company, Flagship and I, at cash in. So the sum of our money was the value of the company. I had I had a third of it, but which I paid for, and Flagship had 2/3 of it, which it paid for. And we had no IP at the time, just ideas. So we funded it, which was the 180K or 200. But every round after that, plus or minus, was a double of the previous round. So seed A, B, C, D, IPO, every round was a double. Annual return, 100% IRR, because we did financings more or less every year. So the initial money was a 32x. The next round was 16x and 8 and a double before the IPO. So what happened was, because of these major step-ups in value, the board kept raining stock options on me. So I wound up, this is a matter of public record, but I wound up owning my family and I owned 14% of it before the IPO.
My goodness! So it was a good story for you. I mean, that's one of the things
Turned out well.
Yes, and clearly your previous experience led you to sort of zone in on the right value that you thought you could continue to have.
That's right. Basically, it was, if you say, so these days IPO values are higher. You know, typical IPO these days might be a 500 or a 400 pre for the kind of companies I'm involved with. But the idea is, if you know what the last private round value is going to be, you can scale back, because if you get overly ambitious on valuation early on, you get hurt later on. Your stock option grants aren't as big as they could be, because you haven't done anything for me. Why should we give you stock, Stan? Yeah. Or it's so you need to leave headroom for every subsequent round. And we did have a comprehensive plan on fundraising of how much money we'd need to raise.
Great. Amazing.
Which was more or less accurate, as it turned out.
Perfect. So we're cruising towards the end here. I think we're probably going to go a couple minutes over, but that's fine. Nobody's going to shut us off. We don't have any networks. People are going to start running ads on us or anything. So I'd like to transition to your post-exact life, and you've gotten involved in a number of companies, and you've incubated, and you started. Can you talk a little bit about the things you've been most excited about post-exact?
Well, these days I'm in my 70s now, and I'm officially an old guy. So I represent a couple of things. One is I'd like to think I'm still pretty energetic and creative, but the other thing is I've been in the diagnostics industry, 70 years old. I've been in it for 35, so I'm kind of institutional memory now, and I have a couple of successes under my belt. So I bring to boards of directors simply the voice of experience. It's good judgment comes from experience. Experience comes from bad judgment. I got plenty of bad judgment to tell stories. So as a board member, I'm wouldn't be useful on the board of a large commercial scale, Medtronic, et cetera, kind of company. But I get involved with startups that either are spin-outs from universities or solo inventors have taken forth. And this isn't a bad business model. Two of these are now unicorns. They're over $1 billion private companies with over $1 billion market caps, or roughly $1 billion market caps. So what these CEOs are is better than me, I should tell you, because if my journeys were, 10 and 20 years, their journeys are 6 and 8 years. Enough of a improvement in quickness to reach the $1 billion market cap point that I take note. So as a board member, what I contribute are, I'm particularly good at I become good, having struggled so much at Cytyc with clinical trials. I really immersed myself into it. I wound up teaching courses at MIT about clinical trials, and spend a lot of time working with management on this. What's a value proposition? A value proposition is something you show with a clinical trial result. If you have a clinical trial result that doesn't support your idea of value, you won't get paid for it. You'll have spent a lot of money and have little to show for it. So connecting the dots between the value proposition and the clinical evidence that supports the value proposition is probably where I spend most of my time in working with CEOs.
Right.
That's a lot of fun. For me, it's a lot of fun. It's not the same as being an inventor, but when I see a light bulb go on about the connection between medical evidence and value proposition, I find that very fulfilling.
A wonderful thing. So, if any of our listeners are going to take one thing away from this, maybe it would be your three core things that you laid out way at the beginning: huge markets, clear value prop, strong IP. Which I have a sense that you still support that that conclusion, and that spending a lot of energy tying together your clinical evidence and your value prop is super high value activity. So I'm going to ask you a couple more questions before you wrap up. So so if you think about your own characteristics, and at the beginning, you didn't probably realize which of your characteristics were going to be key to success. Some of them probably seemed like they're big nuisances, and other things you thought would be important didn't. Can you talk about what you learned about yourself in this process?
Yeah. So I didn't set out in my career to be a businessman, that is to integrate science and business and operations and sales and stuff, but I became that. I'm better at some things than others, but I have learned something I'd like to pass on to you, to all of you. It's that science and business lie on a continuum, on an intellectual continuum. The same analytic skills that make you a good engineer will make you a good business person. Same is true for thinking through clinical problems. Same is true with thinking through manufacturing problems and selling and marketing problems. They're all different kinds of analytic thinking, and they're all creative. Very often scientists, especially those that come out of academia, say, well, I don't get the business part. And that's the sign of a lazy mind, ladies and gentlemen. It's all fun. And I never knew that when I started, Scott. And by trying to act more broadly, I'd like to think it's a step towards an important step to optimizing the outcome.
Amazing.
personally and for the companies and for the people that work in my companies.
That's when I talk to university classes, I kind of talk about that. At the end, we're engineering an outcome for a company, and you're going to be way more important as an engineer if you understand that in your career.
That's right.
That's
right.
brilliant.
Yeah.
So
engineering education do a poor job at teaching the young engineers what role engineering plays in this enterprise, whether it's a public enterprise, a government enterprise, a private enterprise, or a publicly traded private company. It's a capitalist company. We don't teach that, and that's a shame. So I'm glad, Scott, that in your classes you have a chance to talk to the young about that.
Mm-hmm. So to wrap it up, if you were now telling your 20-year-old self something, what advice would you give yourself?
Yeah, so this is a good question, and it's something I do think about because I've got kids in their, plus or minus 40 or so, and I'd say it's that you can learn anything. You don't have to have a degree to learn it. You can't practice medicine without a degree, but you can learn medicine on your own. You can learn as much as you have the capacity for. You'll find plenty of people to support you in your self-taught education, and that's the most empowering thing that I ever did for myself was that couple of year period where I would go on vacation with the medical textbook and the medical dictionary. So rather than, I have to go to medical school if I want to do Pap Smear, I can learn this. I can learn anything. I can learn stuff about new branches of medicine and new things in engineering that I don't know. And if I can do it, you can do it. So I think this idea that there are no limits to what you can learn, whether it's science, engineering, business, sales, and marketing, it's within the span of each of us to do that. And with that empowerment, you can. Without that empowerment, you become less than you can be.
Mm-hmm. Well, this has been completely wonderful. I wish we could talk for another hour or even two, because I've got so many more questions I'd love to ask you. And I know that there's been a number of questions from our audience, and I haven't gotten to very many of them. Apologies to our audience. I will pass those questions along to Stan in case he chooses to respond to any of them. And
let me see. We have quite a bunch. So can I respond to the group as a whole? Because the questions are actually excellent.
You can carry on as long as you want. I was planning to wrap it up, and you could just email these people. But if you want to pick a couple and answer them, that would be just great.
Let me do a couple here. Let's see. They're also quite good. It's actually hard to choose here. Let me take one because I am involved in it. This is from Iran. With recent developments in AI and medicine, do you see the technology ripe to replace humans in analyzing Pap smears? So I'm a founding board member of a company called Path AI, which is, the name implies, does pathology slide reading on a fully automated basis. Today, there are semi-automated AI-like systems for reading Pap smears. They do what we had thought about doing in the 1980s, which is highlight areas of abnormalities in slides, and these are commercially available instruments and from a couple of vendors, including Cytyc, to show candidate abnormal cells. That's not AI. AI is read the slide, come up with a diagnosis, and do better than humans. Well, Path AI is headed down that direction. So I think that this is a question of when and not if. Path AI has a number of serious competitors staffed by serious people as well. So I think this is inevitable for pathology and radiology. And it's not that pathologists will go obsolete or radiologists will go obsolete, but more and more of the routine work will be done by AI, but the overall responsibility still stays with the humans. Here we go. Dan Conley writes, I'm a big fan of robotics and automation and a co-investor with you in Thrive Bio with great optimism. How has the intellectual property landscape evolved to block imaging technologies since your early days with Cytyc? I think, Dan, that it's a hard question. I think that patents expire, and they expire after 20 years. Then they become public domain. I think I am not concerned so much about blocking patents in imaging, especially in the age of AI, when new opportunities for IP are being created, and therefore ways of getting around existing IP, aging existing IP, is possible. So I think imaging remains rife for invention. I'd like to comment here. This is a very important question from Michael M. Thank you for the nice presentation and discussion. As an engineer, you are making innovations. Could you comment on the trade-off between technology push and market needs pull? Thank you. I'm pretty much an outlier. I've never been, gee, this is a cool technology. Let me find, that's the technology push. Let me find an application for it. I do read a lot to this day. And the way I read the New England Journal of Medicine, for example, is, oh, this is an interesting article about an important problem. Did the authors demonstrate that their solution is viable, that it can scale, that it can be medically important? And most of the time, the answer is yes. But every once in a while, the answer is no. And that's a market pull. And that's when I start thinking about, gee, how could I solve this? And as the part of the evolution of Exact, as I told you the story of this lunch Tom Farb and I had, and shortly thereafter, there was an article in the New England Journal about a multi-year trial, a 15-year trial, showing a reduction in mortality in colorectal cancer by screening using something called fecal occult blood screening. It was a pretty good reduction in mortality with a test that was not very sensitive. A careful reading of the article would lead one to conclude, led me to conclude, that the reason the test looked like it worked, it was actually not working at all, but was random selection for colonoscopy. Just take 1 out of 4 people and say, you get a colonoscopy because there was a false positive on this fecal occult blood test, you'd get the same results of reduction in mortality. So that to me was a breathtaking moment reading that. It was the moment when I knew I had it for Exact, that this was a highly cited paper by respected academic authors, and they completely didn't understand their own data. So that was another one of these market pull moments. Size of the market was one. A poor test demonstrated a reduction in mortality. Imagine what a good test could do. So that was that's my view. And Michelle, sorry, it's not Michael M, it's Michelle M. There's no right answer here. This is a personal perspective. John S. wrote an interesting question. Did Exact get a CE mark in the interim 5 years before PMA? I would say this, both my experience at Cytyc and Exact had led me to conclude that US companies, I believe Canadian companies, but certainly US companies, trying to market their products overseas before they have US approval are uniformly met with, why are you trying to sell this to us? Get FDA approval first before we'll buy. We actually started a Cytyc subsidiary in France, and we had almost nothing to show for it because of that reason. So come back after you have FDA. And then the company did years later and was very successful. So Dan C writes, one last one maybe, Scott?
Sure, yeah, one more is good.
I'm involved in a company called Lucid Diagnostics, and Lucid uses an invention, is based on an invention by a collaborator at Exact named Sandy Markowitz for detecting the precursor lesion to esophageal cancer. Esophageal cancer has a very poor prognosis. I think it's 5-year survival is 20%. It's caused as well. What causes cancer? In one sense, it's still a mystery, but there's a strong association between long history of proton pump of reflux, long history of reflux, obesity, which causes reflux, and smoking and excessive alcohol consumption. So there's a high risk population of a fairly large number of people at elevated risk for esophageal cancer. Being diagnosed with it is pretty much a death sentence. However, the precursor stage, which takes, like in the colon and like in the cervix, takes many years to develop, the precursor stage is called Barrett's esophagus. If you find Barrett's by upper GI endoscopy and treat it by ablation, you've cured the patient. and remove the risk of esophageal cancer. So a company called Lucid, I got contacted by them a year ago. I was very excited because it fits my sort of understanding of the pre-malignant, malignant progression and the value of intervening with a pre-malignant at the stage of pre-malignancy very well. The invention, the gizmo, already is FDA cleared, and the company is working on a PMA for the combination of the gizmo and the test. And if the company is successful, it has the same potential as Exact and Cytyc did to be multi-billion-dollar companies because of a focus on early detection and cancer prevention.
Wonderful. Well, there you go. This is rolling along so wonderfully. It's just been a total pleasure. Thank you so much. And this is a gift from thousands of people who watch this presentation over time as well. So once again, it's been a total pleasure. Thank you to our audience for sticking with us. And if you want to watch this later or want to refer to anybody else to watch it, you can just go to starfishmedical.com/webinars. And this will be posted in the next few days.
Great. Well, thank you very much, ladies and gentlemen. I enjoyed speaking with you. And Scott, thanks for hosting.
My pleasure. Thank you.